Hello, my name is Melody Haler. Today I want to first share with you a little about myself, and then I will talk about my project on site-directed mutagenesis of human tubulin genes. I'm a non-traditional student at the University of Wyoming, and I work part-time as an internet search engine evaluator. When I have the opportunity, one of my favorite places is spending time in nature. Previously, I completed an Associate of Arts degree in history with a minor in English. Currently, I am a senior and I plan to graduate this December with two Bachelor of Science degrees, one in biology and one in psychology. In the fall of 2022, I hope to be accepted into a clinical neuropsychology graduate program. I've been a part of the INBRE program through University of Wyoming at Casper College for the past couple of years, working with Dr. Florence Toole-Finley. During this past year, Caitlin Edwards, my lab partner, has joined us. Our project is a collaboration with Dr. Brian Sherrington and Dr. Heather Rothfuss in Laramie, Wyoming. The goal of our research is to determine how the post-translational modification citrullination affects tubulin. Citrullination is regulated by different peptidyl deaminase enzymes, also called PADs, which affect how these proteins function. Defects in human tubulin citrullination contribute to neurodegenerative diseases. To evaluate the effects of tubulin citrullination, we need to generate non-citrulinated human recombinant tubulin subunits by insect cells. Our goal was to perform site-directed mutagenesis to generate two mutant tubulin genes, alpha-1 with 1,356 base pairs and beta-3 with 1,353 base pairs. This removes the citrullination target in the peptide sequence. A number of steps are required before site-directed mutagenesis. I've not yet reached that stage, but I will share some information about the techniques I have used. The plasmid PFAS-Bac dual PFBD expression vector contains both alpha and beta tubulin genes. I've been working on subcloning tubulin alpha-1 gene from the PFBD expression vector into the smaller cloning plasmid vector PUC19 to isolate it for future mutagenesis. This prevents accidental mutagenesis of the expression vector or beta tubulin gene. The tubulin alpha-1 gene is excised with two restriction enzymes, EcoR1 and HinD3, which will be used to prepare PUC19 to receive the tubulin alpha-1 gene. Shown here is the first step in subcloning by culturing bacterial clones to the plasmid DNA PUC19 and PFBD alpha-1 and beta-3 tubulin. Since these vectors carry ampicillin-resistant genes, the bacterial clones are cultured in the same condition using LB ampicillin medium. Then they are inoculated with frozen clone stock and finally incubated. After this, we performed plasmid DNA extraction using an eZNA plasmid minikit 2 from Omega Biotech. This involved alkaline lysis of the bacterial cells, binding, washing, and elution of the DNA. We confirmed recovery of plasmid DNA through 0.8% agarose gel electrophoresis using 1x-TAE as a running buffer. The left picture shows the gel at the start and the right shows it after DNA separation. Each sample in the gel has 1x loading dye, sterilized deionized water, and plasmid DNA. Then the gel was stained with diamond stain, an intercalin agent that wedges itself in between the bases of DNA fragments, allowing the DNA to be detected under UV light. On the ChemiDoc, using Bio-Rad Image Lab software, we determined if plasmid DNA was recovered. If so, we proceeded with restriction digestion of the plasmid DNA. Our plasmid DNA required two cuts, the first with enzyme EcoR1, and once that was successful, the second with enzyme HinD3. This required an overnight bath at 37 degrees Celsius. Then agarose gel electrophoresis was performed once again, and once this was successful, I was able to begin electroelution. This looks similar to typical agarose gel electrophoresis. Only the outer left and right sides of the gel are stained, making the migration profile visible to locate the DNA fragment in the unstained middle part. The whole gel is not stained because intercalins like diamond stain are mutagenic compounds that would then need to be removed from the purified fragment before cloning. The partially stained gel was reassembled on the UV box to locate and isolate the DNA fragment to be cut from the unstained middle portion. Next, the cut pieces of gel were placed into dialysis tubing with a small volume of 1x-TAE buffer. These were then placed in the electrophoresis tray once again to subject the DNA fragment in the gel to an electric current to facilitate DNA migrating out of the gel slice into the buffer in order to recover it through ethanol precipitation. I recovered the released fragment by pipetting 1x TAE buffer from the bag and then ethanol precipitation after the salt addition. The DNA sample is pelleted through centrifugation and washed by the addition of 70% ethanol to remove excess salts. Another centrifugation step allows recovery of the clean DNA pellet, which is then dried under vacuum in a speed vac before resolubilization in sterile distilled water. Recovery is then checked through agarose gel electrophoresis. The next step will be recovering the gene and vector for both PUC19 and PFBD. Experimental work and laboratory interactions were delayed due to the COVID-19 pandemic. I've been able to linearize both PUC19 and PFBD with HENDI3. The gel on the left has an arrow pointing to linearized PFBD, and the gel on the right has an arrow pointing to linearized PUC19. Although I have not been able to complete the process of site-directed mutagenesis, I have appreciated being in the INBRE program, learning multiple molecular techniques, and learning how much patience and stamina is required in scientific research. I've enjoyed working with Caitlin Edwards and Dr. Florence Toulé Finley, and I appreciate everything they have taught me.